Our guest today for NKDO YouTube channel series of discussions with transplant industry experts is Dr. Suzanne Illdstad, the Chief Scientific Officer and founder of Talaris Therapeutics, a NASDAQ-listed medical research company which is engaged in phase 3 and phase 2 trials, which have the potential to transform the solid organ transplant industry, as well as certain autoimmune conditions. Dr. Illdstad has received numerous awards and honors, including election to the National Academy of Medicine and National Academy of Inventors, as well as the Mayo Clinic Distinguished Alumnus Award. She has authored over 230 manuscripts and is a named inventor or co-inventor on over 100 patents. Dr. Illdstad, you are our first interview with an expert in the venture stage of developing therapeutics and transplant, and we are interested in learning about both your background and the current state of development of FCR-001 at your company. You have a particularly good video on your website, which I would like to show to set the stage for our discussion. But before I do that, I ask that you provide the context for that video with some background on your career and how you have gotten Talaris to this level in its development.
I'm a transplant surgeon by training, and I saw firsthand the challenges that transplant recipients face after they receive their transplant. The immunosuppressive drugs that are critical to maintaining the graft also cause a lot of toxicities. They cause cardiovascular disease, an increased rate of malignancy, increased susceptibility to infection, and ironically, they actually damage the kidneys over time, so that people who are on long-term immunosuppression may lose their kidney from the toxicity of immunosuppression. And so my passion became to identify a way to overcome the need for the immunosuppressive drugs. And it had been known for about 50 years that if the bone marrow stem cell from the donor takes, it re-educates the recipient to tolerate the donor and not try to reject it. And so we worked on finding a way to make bone marrow transplant safe enough to be able to induce tolerance.
Okay, that's a great explanation. Now let's watch the video. I'm going to share my screen.
Immune tolerance is when the immune system does not mount an immune response to antigens it recognizes as self. However, lack or dysfunction of immune tolerance can be detrimental in settings such as allogeneic transplantation and autoimmune disease, where antigens deemed as non-self are attacked by immune cells. What if there was a way to induce tolerance in these patients so antigens are instead recognized as self? There are 3 broad applications where this could be advantageous. We will focus on allogeneic transplantations. Kidney transplantation is a life-saving procedure for patients with end-stage kidney failure that is caused by a number of conditions. The greater the degree of HLA matching between the donor and recipient reduces the risk of the recipient's immune cells from recognizing the donor cells as non-self, lowering the risk of organ rejection. However, even in a 6 out of 6 HLA match, kidney transplant recipients still must take lifelong immunosuppressants to prevent rejection, which itself has many drawbacks that can adversely affect transplant outcome. Development of the immune response begins with recognition of HLA by T cells. T cells differentiate into effector cells. Effector T cells, derived from the host, can injure the transplanted kidney, a process called acute rejection, leading to graft failure. Reprogramming a transplant recipient's immune system to durably tolerate the kidney could circumvent these drawbacks. FCR-001 is an investigational cell therapy that aims to facilitate allogeneic tolerance by creating a dual immune system in the transplant recipient. 3 weeks prior to the kidney transplant, mobilized stem cells and immune cells are collected from the peripheral blood of the kidney donor. Talaris processes hematopoietic stem cells, facilitating cells and alpha-beta T cells from the collected peripheral blood, which make up the FCR-001 product that will be infused into the kidney transplant recipient. 4 days prior to the kidney transplant, the kidney recipient undergoes non-myeloablative conditioning to make space in the bone marrow for the engraftment of kidney donor HSCs. The recipient receives the donor kidney on day 0. 1 day following the kidney transplant, the FCR001 is infused into the kidney recipient. Facilitating cells are believed to enhance the homing and migration of donor HSCs to the non-myeloablated bone marrow, where they engraft next to the recipient cells. Donor and recipient HSCs mature into mutually tolerated functional immune cells. Donor facilitating cells, along with a specific number of alpha-beta T cells, may mitigate the risk of graft versus host disease. Donor and recipient immune cells are released into the peripheral blood and travel to the kidney. This durable chimerism means the donated kidney is recognized as self, irrespective of the degree of HLA match between donor and recipient. This reprogramming of tolerance lowers risk of immune-mediated rejection. This can lead to discontinuation of immunosuppressants and avoidance of debilitating treatment side effects. FCR001 has several potential clinical applications for induced tolerance through reprogramming, restoring, and replacing in allogeneic transplantations, autoimmune disease, and blood disorders.
So, Dr. Illdstad, I don't think anyone with even a passing knowledge of the issues in transplant could watch that video and not get excited about the potential value of FCR001. In listening to the narrative, however, the words may and could come through loud and clear. Please give us a sense of where you stand in the development of this therapeutic and what is required to get from this stage of development into an approved clinical application.
For drug development, the FDA has different classifications, and you start with, there's phase 1, phase 2, and phase 3. You start with phase 1, which is safety, go to phase 2, which is proof of concept, and then 3, which involves a multi-center site to show that it works at more than one place, and then it goes on to approval. We transplanted 37 patients in our phase 2 for the living donor kidney, and showed that we could achieve withdrawal of immunosuppression in 26 of those 37 patients. And we have patients as far out as 12 years after their transplant who have been off immunosuppression and have had no evidence of rejection, no evidence of deterioration of graft function, and who are living essentially normal lives. And now we're doing the phase 3, which is a multi-center study involving 17 sites.
So what is the success rate of your approach in the phase 3 thus far, and how do you measure that success rate? Any details you can provide on the measurement process would be helpful.
The phase 3 has just really been enrolling now. And in a number of the patients that we've done a small number because of the challenges of COVID. And in the small number that we've done, we've had 100% have engrafted. And we're in the process of weaning them from immunosuppression. In the phase 2, we refined the management of the patients and really optimized the cell dosing. And in the last 17 patients that we did, we had 82% success of establishing durable donor chimerism and being able to wean the patients off immunosuppression.
What are the characteristics that make one patient a good candidate for this protocol and another one not a good candidate?
We have what are called inclusion and exclusion criteria. One of the major exclusion criteria are patients who are highly sensitized. So if someone has antibody against the donor, obviously they can't have a kidney transplant. Or if they have a very high panel reactive antibody, which is called a PRA, which is reflective of sensitization, greater than 80%, they're excluded. And also body weight is a criteria. age is a criteria. We have a span of age that's acceptable. And the best place to access all that information is on clinicaltrials.gov. Our study is registered there, and it lists all the inclusion and exclusion criteria. And I think our website has some detail on that as well.
Okay. I've heard that this protocol may increase the risk of graft versus host disease and the patient mortality risk. Was this a phase 1 or phase 2 issue that you've resolved?
We had 2 patients develop graft versus host disease in the phase 2. And one was treatment responsive, and the other was diagnosed very late after development. The patient waited to go to the hospital and then went to a local hospital that was not a transplant center. And by the time he was diagnosed, it was very aggressive, and he also had CMV colitis. So he was a very complex case. What we've done in that case after that is to have one of our nurse coordinators contact patients weekly and remind them if they get any symptoms at all, whether it's infection, fever, potential GVH, that they go to a transplant center of excellence and be seen right away. And that seems to have helped a lot.
Terrific. Okay. In trying to understand how the components of FCR-001 work together, it appears that the magic sauce is what you call facilitating cells. That's a new concept for me. Can you please tell us what they are and how they function?
We used a mouse model actually to identify what cells from bone marrow are needed to help the stem cell engraft and mismatched recipients. And we found a unique cell that is not a stem cell, that has unique markers. It's CD8 positive and T cell receptor negative. And it allows engraftment without graft versus host disease. It's a living cell. And so it has, not surprisingly, multiple functions, we believe. It enhances homing and migration of the stem cell to the niche, and it induces a very tolerogenic microenvironment. We know that it induces a tolerogenic cell called regulatory T cells in vivo, and it's a self-perpetuating effect. And we're still defining further the mechanism of action.
I see that in addition to the phase 3 trial for the FCR-001 for prospective recipients, you're also running a phase 2 trial for existing transplant recipients. Can you talk how this trial is set up? How many recipients are included? Is the trial closed, or is there room for additional candidates? How do you measure success or failure in a trial of this type?
We're actively enrolling now, so it's an interesting story. One of my colleagues' patients at Northwestern had a kidney transplant, and she brought her donor and said, have you ever considered trying to induce tolerance after the fact, after the kidney transplant, which is a brilliant suggestion. And so we essentially used a rat model to show that indeed we could do the kidney transplant first and then establish chimerism and tolerance after the fact. And that's what the phase 2 delayed tolerance is. And we are currently actively enrolling patients where we want to start. We don't know what the effect of having the kidney in place before inducing tolerance has on immune activation. So we're starting right now with patients who have not had a kidney transplant a long time ago, but who have had one within the past few months. And then we'll inch further and further after that in terms of the duration between the transplant, the kidney transplant, and establishing chimerism to see where we can induce tolerance and if we need to modify the conditioning at all. So that's why it's still a phase 2, but we're definitely recruiting patients right now.
NKDO has been involved in matching donors and recipients for the last 7 years. There's no shortage of candidates we could provide you if you'd like some.
That would be great. No, that would be wonderful.
The prospect, how long do you think you'll be in phase 2, and when might it move to phase 3 if it's successful?
It's hard to know. Probably if we have 5 or 6 consecutive successes, I think we would start considering moving to phase 3. Normally for a phase 2, you don't need a huge number of patients if you can show efficacy and safety.
Wow. So this is something that could be very interesting for folks who've had a transplant and are living life on these immunosuppressive drugs. So how would you measure success or failure in this kind of a trial?
In the same way that we do for the Freedom 1 study, the living donor kidney simultaneous. So we continue the patients on their standard immunosuppression for about 6 months to give the donor graft a chance to settle in, the FCR001. And then we start to wean, we test for chimerism. And if we see chimerism greater than 50%, meaning the donor cells circulating in the recipient's blood. And if we do a biopsy and see normal histology on the biopsy, and if renal function is stable, we will first stop the mycophenolic mofetil and continue the tacrolimus, but taper it down over the following 6 months, and then do another biopsy and check for chimerism. And if everything's a go, then we stop immunosuppression.
Wow. Okay.
So it's very objective criteria. And
Yeah.
we now know that if we see chimerism within the first 3 months, that we're virtually, it's predictive of being able, if it's greater than 50%, predictive of being able to wean the recipient off immunosuppression.
That is very exciting. Okay. Your second of 3 programs addresses the problem of severe autoimmune disease, employing a treatment you call facilitated allo-HSCT therapy. Please explain what that is and how it works.
That's really the same product as the FCR001 and what we're using also in the delayed tolerance. Your immune system is generated from your stem cell. And essentially, when it was observed in patients with life-threatening autoimmune disease who also had leukemia or hematologic malignancy, that if they had an allogeneic transplant, that they often were cured of their autoimmune disease. And additional studies were done, including our own, in mouse models of autoimmunity to show that that was indeed the fact, that you can actually, if you transplant a healthy immune system into someone with a misbehaving immune system, which is really stem cell derived, you can make the abnormal immune system behave. And so we decided to start with life-threatening scleroderma because that's a very high unmet need, and there really is no curative therapy out there. And that's another study that we're actively enrolling patients in. We have 2 sites that are involved. And again, that the criteria for all of that is listed on clinicaltrials.gov.
Okay. Is lupus a disease that might benefit from this therapy?
Yes.
Wow. Okay.
There are about 60 autoimmune disorders, and many of those in humans now have been shown to be had remission induced by bone marrow transplant in patients who had hematologic malignancies and had a bone marrow transplant for that. So there's a great deal of clinical data to support that.
That's very exciting. Your third program addresses severe non-malignant blood, immune, and metabolic disorders. If I understand the description on the website, the problem you are seeking to solve is a reduction in the severity of the protocol heretofore necessary to treat these disorders using what you call a non-myeloablative conditioning regime. That's a tough one. Can you talk about what that regime entails and how the phase 2 trial is proceeding?
We haven't started that study yet, but again, if you can safely transplant bone marrow from a healthy donor, you can treat a number of different immune disorders that patients experience. And it used to be thought that you had to completely wipe out the recipient's own immune system with lethal irradiation, lethal conditioning. And that was the goal for leukemia because you want to get rid of all the leukemia cells. But it turns out that you can do a very reduced conditioning regimen that can essentially be performed as an outpatient rather than being in the intensive care unit for a month, and achieve the same outcomes, especially in non-malignant disease. And so the reduced intensity conditioning is something that we actually developed in the mouse, that the Fred Hutchinson Cancer Center scaled up to the dog and then started doing humans. And they found with a very low dose of radiation and a low dose of myelotoxic drugs that they could establish chimerism and do it on an outpatient basis. And so it's a very different landscape and risk-benefit ratio if you can do the non-myeloablative conditioning.
Okay, well, all three of those projects are very, very exciting. I do have one question for you, and that is there any prospect for naming FCR001 something that's a little more user-friendly?
Probably not now. There will be a name once it gets approved for commercial. There will be a name assigned to it, and it has to meet all sorts of criteria that the WHO and the regulators have. But
right. So
yes, eventually, but it might even be more complex. You know, some of those monoclonal antibodies that are out there have very complex names.
All right. You've been very, very kind with your time. Before we sign off, is there anything that I have not asked you that I should have asked you that might be informative to our viewers?
No, I don't think so. You know, I think that I would encourage anybody interested to reach out either to the Talaris website or go on clinicaltrials.gov, and all of our protocols, our trials are listed on that with the inclusion and exclusion criteria.
Well, I can tell you that the Talaris website is very well laid out and provides a great deal of information. So I would encourage anyone who has interest in learning more about this to start with their website. Dr. Illdstad, I want to thank you for your time and helping us understand the exciting new medicine you have pioneered at Talaris Therapeutics. We wish you continued success, and we hope to revisit this discussion down the road as these protocols mature. Thank you.
Well, thank you very much, and thank you for the work that you do.